When we think of a sugary drink, it's usually in the context of taste, refreshment, or a quick energy boost. A cold soda on a hot day or a sweetened iced tea with lunch rarely seems like a life or death decision. Yet the latest research in Nature Metabolism uncovers a far deeper story, showing how these beverages can play a covert role in accelerating cancer spread. The study highlights the molecular journey that begins with a simple sip and ends with cancer cells gaining the fuel and mobility they need to metastasize, with a central player in this process being the enzyme SORD. Imagine your body as a bustling city. Cells go about their daily routines, maintaining order and structure, keeping the traffic of nutrients and signals flowing smoothly. Cancer cells, however, are like troublemakers who want to leave their neighborhood and invade other districts. They need energy and guidance to do this, and sugary drinks unknowingly provide both. Fructose and glucose from those beverages flood the system and interact with SORD, which functions like a turbocharged engine. Normally, SORD is a helpful enzyme, quietly converting sugar into energy for routine cellular processes. But in the presence of excess sugar and cancer cells, it shifts roles, powering these rogue cells like a high-octane fuel system, giving them the ability to move faster, adapt, and ultimately metastasize. A casual sip transforms into a molecular accelerator that propels cancer along its journey, turning the body's own metabolism into a pathway for disease progression. Yet this story is not one of hopeless inevitability. On the contrary, the clarity provided by this research brings optimism and opportunity. By identifying exactly how sugary drinks interact with SORD to enhance cancer mobility, scientists now have a concrete target for intervention. Therapeutic strategies could be designed to inhibit or regulate SORD's activity, effectively cutting off the fuel line cancer cells depend on. This represents a shift from generic treatment approaches to precision strategies, ones that attack the problem at the molecular level while leaving healthy cells largely unaffected. Understanding the mechanics of the journey makes it possible to intervene, redirect, or even stop it entirely. At the individual level, this research translates into actionable knowledge. Limiting sugary drink consumption is not just about weight control or managing blood sugar, it's a way to reduce the energy available to SORD-driven cancer cells. Opting for water, unsweetened beverages, or whole fruit instead of sweetened drinks is a proactive step that directly impacts the molecular environment in the body. Each small choice contributes to a larger defense, slowing down or preventing the sweet fuel journey that cancer cells might otherwise take. Awareness becomes a tool, transforming everyday decisions into forms of prevention that are within anyone's control. Importantly, this study also highlights nuance and hope. SORD itself is a normal and necessary enzyme, essential for healthy metabolism. The problem arises only when excess sugar and cancerous conditions align. This distinction is powerful because it shows that biology is rarely black and white. It demonstrates that interventions can be targeted, intelligent, and strategic, rather than relying solely on fear or broad restrictions. It gives both researchers and the public a clear path to reducing risk while understanding the context in which the risk arises. Ultimately, From Sip to Spread reframes the conversation around sugary drinks and cancer. It shifts the narrative from vague warnings to a precise understanding of molecular dynamics. We now see that every sip carries potential consequences at the cellular level, but we also see that knowledge gives us control. Awareness, moderation, and informed lifestyle choices become powerful tools, and scientific insight provides a roadmap for therapies that could one day disrupt this pathway altogether. What seemed like a simple indulgence, a sweet drink, becomes a story of opportunity, the chance to understand, prevent, and intervene, turning a potential threat into a catalyst for action, hope, and better health outcomes.